my pleasure to be able to introduce our speaker for today, Dr. David Gillespie from the Pacific Agricultural Research Center in beautiful Agassiz, British Columbia. And for those who've been up there, it has been an absolutely beautiful station. I was very lucky about 20 years ago. I spent a short sabbatical with Dave and had a wonderful experience there. So, if you're ever up in that area, it is really spectacular. Dave is a native Canadian. Obviously, actually spent most of his time in British Columbia. He has both a Bachelor of Science degree in biology, a Master's in biology, and his PhD in biology from Simon Fraser University. So he didn't go too far away, basically, and now he's at Agassiz, where he is a senior research scientist. So Dave's area primarily is biological control almost exclusively. He does work in sort of field systems with classical biological control. He has a large project now on canola, for example. But his major contribution really is focused on biological control in greenhouse systems. For those that know British Columbia, the greenhouse industry there is huge. The vegetable industry especially, so Dave has made remarkable progress in advancing biological control in vegetables, not only in BC, but literally around the world. And so what Dave has done is really, investigating sort of native natural enemies, understanding them, and basically show how they can be used in greenhouses. And so because of that effort, a lot of what we deal with now on a commercial basis, in other words, what's available commercially, internationally, really came out of Dave's lab. So predatory mites, some predatory cystidiomidae, and especially the predatory murids. A lot of that work, a lot of biocontrol that goes on in California with those natural enemies, actually, the foundation of that work has come out of Dave's lab. And not only has he developed new natural enemies, but sort of the delivery system. So, for example, a lot of how we use miridae, predaceous miridae in greenhouses, how we do that really has come out of that Dave's lab. He was elected as an honorary member of IOBC, I think, because of his work. And today he's going to be talking about a broader topic, that is the influence of climate change on biological control. He is also an adjunct professor at Simon Fraser, works very closely with Bernie Reutberg, that many people know. And I think Bernie's a little bit of inherent in this presentation.
Oh, great. Thank you, Michael. It's a tremendous honor and pleasure to be down here, and coming from British Columbia, where the temperatures are running in the 40s. If I'm lucky, daytime highs and it's pouring rain. This is lovely weather, 20 degrees, and no rain. It's a real treat. So yeah. Well, you've been hoping for rain, so maybe you'll bring it with me. Yeah. So this what I'm going to talk about today sort of stems from work that I started a number of years ago on biological control of aphids in greenhouse peppers, and we developed this little predator-prey parasitoid host system with green peach aphid that we were starting to try and tease apart. Why does it work? How does it work? When an opportunity came along to start to think about in the context of perturbations and the influence of high temperatures on this system. So what I'm going to talk about today is some of that work, where it started from what we found initially, and then some studies that are very much still in the wet ink stage. I've just finished collecting the last little bit of data. In fact, my technicians are still processing some of the samples today, so it's going to be very recent work, which is very much unfinished, soggy clay, as it were. Biological control in the face of climate change. Everybody's presumably seen this graph or an iteration of it. Carbon dioxide levels in the atmosphere are changing. They're changing dramatically in post-industrial revolution time. And as a consequence, we see this phenomenon called global climate change. An increasing concentration of carbon dioxide, which is leading to changes in ocean pH, changes in global average temperatures, changes in wind patterns, changes in rain patterns, and so on and so forth. And there's in the public consciousness, there's this global trend towards increasing temperature. And every time you get a cold day, you get the naysayers sticking up their hands. But those increasing global mean temperatures are changing the world around us in a fairly profound way. We're seeing changes in distributions and ranges of animals, changes in seasonal activity, synchrony between parasitoids, hosts, flowers, and pollinators. We're seeing changes in overwintering survival. So we're seeing a whole host of things happening as a result of this increasing mean global temperature. But there's another trend underlying that, which is that as that global mean temperature changes, there's an increasing tendency towards increasing variation around that mean temperature. So you're getting greater and greater extremes of temperature. And the models tell us then that the future is going to hold a future with increased mean temperatures is going to hold the increased probability of heat waves. The heat waves will be longer, they will be more frequent, and they'll be more intense. And then that begs the question, well, why do we care? And the simple answer is, in part, in this universal relationship between temperature and a host of life history and biological phenomena. Here I've just graphed development rate as a function of temperature, something every entomologist is familiar with. We have a lower development threshold on the x-axis, a temperature below which the process doesn't take place in net effect. And then as temperature increases, the rate of the process increases to some upper threshold, at which point it's going about as fast as it possibly can. Continue to increase the temperature, and we reach an upper development threshold, a point above which the process, whatever it is, doesn't take place in net effect. If you think about that, extend that x-axis, you have a lower critical temperature, that is a temperature below which the phenomena, the organism freezes or dies from accumulated cold injury, and then at the upper end, around 40-something degrees, you have an upper critical temperature, a temperature at which enzymes denature, the animal dies. So those relationships are really, really important in biology. I've showed development rate. Keep in mind, and so this range in here between the upper threshold and the upper critical temperature is the one that's really important in the context of heat waves. Getting ahead of myself here. So that's the zone in which you see accumulated physiological stress. You see accumulated damage. You see organisms starting to change their behavior in response to these threatening conditions. So there's a lot of stuff taking place in this zone, which is the temperature at which we see, which is of concern for these heat wave environments. And I mentioned, showed development rate. Walking speed increases as a function of temperature. Predation can have a neat little curvilinear function as a result of temperature. So all kinds of things change in an organism's biology as a function of the temperature at which it's foraging at. Body size. And that's a well-known relationship. As temperature increases, body size tends to increase, again, in some kind of a curvilinear fashion. That's great, but smaller organisms eat less. So we've been studying this phenomenon of heat waves and high temperature effects in this community that I've been working with, Aphelinus abdominis. So it's based in sweet pepper. It's a greenhouse crop I work with. Green peach aphid as the host, and then two parasitoids, a Braconid, Aphidius matricariae, and an aphelinid, Aphelinus abdominalis. And so just in case you're not completely familiar with this system, green peach aphid takes 7 to 8 days to go from immature first instar to adult or viviparous. It's a great little animal to work with. The parasitoid, Aphidius matricariae, takes 12 to 14 days, lays an egg inside the host. The larva kills the host, forms a mummy shell of the dead aphid, and emerges, as I say, 12 to 14 days later. Aphelinus abdominalis, in contrast, takes 20 to 25 days, a little bit longer to develop, and has more or less the same biology. It's an internal parasitoid. Our general approach in the early work was to use pepper plants in perforated self-draining bags. So these are confined little experimental units to pop these inside growth chambers at different settings and to conduct 21-day experiments. I'll stop wandering around a bit. So the temperature, average temperature, the temperature at which we conducted all of these initial experiments was 25 degrees on a 24-hour average. Let me show you what that means. So what we did is we took the heat wave and we dissected it into either low frequency, that is just sort of a hot days every twice in a 7-day cycle, or hot days every day. It's a heat wave environment. And we varied the amplitude of the heat pulse, either 32 degrees as an upper maximum, or 40 degrees as the upper maximum. And all of these plots have 25 degrees as the cumulative 24-hour average. So everything was conducted at the same average temperature. Oh, and I will just go back for a second just to bring your attention to these little colors. Throughout the presentation, I tend to use the blue colars as the cold environment and hot colors as the hot environment in the various graphs that we were talking about. We were anticipating we would see community effects. This is the plot for green peat development rate, plot for green peat shape at over temperature. Its upper development threshold is about 32 degrees. The Phidias matricuriae, the theoretical plot extends a bit above 32 degrees to some kind of an upper theoretical development threshold. Aphelinus abdominalis, the blue line, is a little bit more sensitive to temperature and has a somewhat lower upper developmental threshold. So what did we find? In these heat wave environments, plant growth in the absence of aphids was reduced by thermal stress. So only in the low temperature, low frequency environment did we see those plants had slightly more leaf area than the plants that were growing under these more damaging or higher temperature environments. So we saw some effects on plant growth by themselves. Aphid numbers, again in the absence of parasitoids, increased less quickly in the heat wave environments. Keeping in mind this is a log to the base 10 scale, so that's actually a fairly dramatic effect on aphid numbers in the absence of parasitoids. We saw that the proportion of alates in the population was reduced by heat waves, independently of crowding. So as crowding increases, aphids generate alates, winged individuals, which then are the dispersal units that move away from the crowded conditions. And there's a well-known relationship between crowding and the proportion of alates in the population. However, if you do that regression and take the residuals, what's left over is the effect of the heat wave. And under high temperature, high frequency conditions, there was a much lower proportion of alates in the aphid population. As far as parasitoids go, the Phidias matricuriae, numbers of Phidias matricuriae were lower under the heat wave environment, so we had fewer Phidias matricuriae in the system where they were present. We also saw that aphid numbers were reduced by parasitoids in those heat wave environments, but only when the two were present, not when either was present alone. So kind of an interesting effect. And again, this is the shape of the structure of the community is actually changing as a result of this heat wave. But there was no effect of the experimental heat wave on the trophic cascade. So that is, plant size didn't change. So the effect of the reduction of aphid population by the parasitoid wasn't evident in plant growth by the end of our 3-week study. So it's an accumulation, sort of a subset of some of the interesting effects that we saw in this set of experiments that we did. And as biologists, we look at these big, somewhat holistic experiments, and we scratch our head and we say, well, what's happening? Why are these effects emerging from this experiment? And this one's fairly predictable. So if you think back to this development rate plot that I showed you, it makes a lot of sense that Aphidius matricariae should show an increased days to development under the highest amplitude, highest frequency temperature regime, but not under the other temperature regimes. Conversely, Aphidius Aphelinus abdominalis, the development rate of this species was increased under both of the high frequency temperature regimes, irrespective of the peak temperature. And again, that's just reflecting these differences in upper development threshold. And so these differences then become very important. They're part of the driver of the structure of this community as daily temperatures increase. Temperature and mortality were affected. We did some studies using block heaters where we put individual insects into Eppendorf tubes in these block heaters, and then conducted a series of time by temperature experiments. What we found was kind of interesting. So this is temperature on the one axis, exposure time on the other axis, and so Aphidius matricariae as mature larvae inside aphids. So these are aphids that are about to be killed. So they're still walking, but you can see the parasitoid larvae inside them, remarkably resistant to high temperatures. We had to push things up to 44 degrees for at least 30 minutes to start getting any appreciable mortality in the parasitoid population. That's pretty extreme. Similarly, for mummies that were fully formed, 24 hours old, so the aphid is dead, the parasitoid is starting to pupate, even more resistant to high temperatures, 44 degrees for 2 hours, and we still didn't produce 100% mortality in the population. Green peach aphids, by comparison, are complete wimps. Even 38 degrees for 30 minutes or so is producing an appreciable level of mortality in the population. So differential mortality, I'm not saying that the heat wave effects were producing mortality in the population, just that the threat of mortality is there and that the animal would have to do something about it to avoid dying. Remember, confining an animal in an Eppendorf tube and cooking it is very different from letting it on a plant, letting it wander around. And I'll show you why that is so. We saw some behavioral effects. Pupation sites appeared to change with Aphelinus abdominalis. What was happening is that under the high amplitude, high frequency regime, so the hottest temperature every day, Aphelinus abdominalis was shifting its pupation sites off of the plant and onto the soil surface, onto the top surface. So they were actually moving off of the aphids that contained the parasitoids were moving off of the plant under high temperature exposure. We're still kind of working at that one, and I still don't understand who's in the driver's seat. Is it the aphid or is it the parasitoid? Which is why that manuscript is still in preparation. Finally, in terms of these proximate effects, keep in mind that the plant is always an active partner in the temperature exposure. So this is the blue line. The green line is our leaf temperatures. The blue line is the air temperature. And you can see that as the temperatures reach the 30, 32 degree threshold, the plant starts to respond and to actively cool its loose surface. So it's losing water, presumably, to achieve a cooling effect. So the leaf temperature in those experiments is actually quite a bit lower than the air temperature. And similarly for the 32 degree, the low amplitude regime as well. So the plants are affecting some cooling, which then gives the aphids, particularly, an opportunity to move within the plant, to shift position, to seek places on the plant that are coolest. So it gives aphids opportunities to seek refuge. So these experimental heat waves that we were working with, it simultaneously affected plant growth, aphid population increase, caustic disturbance, aphid A-link formation, parasitoid population increases, development, host and parasitoid mortality, and mummy formation. So there's a whole bunch of things that are going on simultaneously. You can't just tease out one effect and say this caused it or that caused the effect. It's a whole host of responses, very complicated responses. So how do we start to think about this? Well, this is some work that Jacques Brodeur and some of his students did in 2012, showing the effect of temperature on the x-axis on two components of functional response. This is handling time, which decreases as a function of temperature as the animal becomes better able to digestion processes increase, the ability of just straight up consumption processes change, and conversely, search rate increases as a function of temperature as the animal moves more quickly and encounters prey more frequently. So there's two very important changes happening just in the context of a functional response. So heat waves affect all aspects of a predator-prey interaction. This is just a standard Nicholson-Bailey plot for those of you not familiar with it. The number of individuals that you're going to produce or in some time interval is a function of the birth rate minus the effects of crowding carrying capacity, birth rate as affected by carrying capacity and effects of crowding, minus deaths, which are a function of crowding and a function of predator density, which is similarly generated by these linked equations. So, great. We really should be writing these equations this way, which is to say that every rate in that equation, everything changeable is a function of temperature. So the generation of individuals at some interval t is going to depend very much on the temperature during that interval. Common sense. So let's just backtrack on heat waves. Heat waves have three dimensions. They have amplitude, frequency, and autocorrelation. So this is a heat wave in Agassiz. Anything above 25 degrees Celsius is a heat wave. It's a pretty wimpy heat wave, but it's what we've got in my region. So if you look at this plot of the maximum daily temperature across time, you can see that the amplitude, the maximum temperature above this 25-degree threshold, which defines our heat wave, it varies. So there's amplitudes, different amplitudes. Hot days occur with different frequency. So in some periods they're rare, in some periods they're more common. And then there's autocorrelation, which is the tendency of one hot day to follow another. So if you have a hot day, weather patterns just suggest that the next day will probably be hot as well. You tend not to get completely random fluctuations in temperature. So a graduate student who's working with Bernie and I, Jordan Bannerman, did some very, very cool work and developed a very complicated simulation. This is just a Leslie population, Leslie model, matrix model. And what he did is a lot of the transitions in this model are generated by temperature, essentially by the Nicholson-Bailey type plots that I formulated that I showed previously. And he ran this population simulation with temperature under 27 different combinations of amplitude, frequency, and autocorrelation of heat waves. So low, medium, high, low, medium, high, low, medium, high. It's a very complicated simulation. This is just an example of his an intermediate range amplitude against 3 levels of frequency. You can see low frequency heat waves, higher frequency hot days, and then autocorrelation on this axis where days tend to be a bit random, days are much more closely associated with each other, and so you tend to get hot days after hot days. So he ran these 27 simulations and generated a lot of populations. This is the population plot for that same simulation, where populations either persisted or began to go to extinction, as is quite often the case with Nicholson-Bailey type model structures. And what he found, which is really cool, which is that when maximum daily temperatures were below an organism's developmental thresholds, frequency and autocorrelation had no effect on community persistence. So low, medium, high frequency, white, gray, and black are the levels of autocorrelation, so no effects. But as soon as you start to exceed the temperatures, maximum daily temperatures were over the organism's threshold, we started to see increasing frequency and autocorrelation of hot days, decreased community persistence in this model. So populations tended to go extinct, local populations tended to go extinct when exposed to high frequency, high autocorrelation, high amplitude heat waves. Okay, too badly here. So let's just go back to this notion of global climate change. So I've been talking about heat waves as an important component of global climate change. But how do we think about this when you think about the context of future growing seasons? So we'll have longer lasting, more frequent, more severe heat waves. We're going to be looking at temperatures that are anywhere from 2 to 4 degrees higher on average during the growing season. And we're going to be looking at carbon dioxide levels that are substantially higher. We're running at about 400 parts per million in the atmosphere. We're looking at a 50% increase in about 40 years to something in the order of 600 parts per million. That's going to have some big effects on agriculture. We're looking at faster growth rate in plants. Carbon dioxide is one of the engines of photosynthesis, as it were. And any plant physiologist in the room is probably ready to throw a brick at me. We will see decreased nutrition available in plants as the carbon-nitrogen ratio shifts. This effect is very well documented. As carbon dioxide levels increase in the atmosphere, plants are allocating relatively more in their tissues to carbon, and so the plant tissues just contain relatively more carbon than they do nitrogen. And if you follow Hairston's green world hypothesis, nitrogen is limited in terrestrial ecosystems. Generally speaking, carbon dioxide tends to increase defense compounds. Some pathways are upregulated, some pathways are downregulated. It's really complicated. Increases in temperature, average growing season temperature. You're going to be looking at faster growth, development, and reproduction rates in all species. Walking speeds of predators will be faster, but prey are going to be escaping at a much higher rate as well. Body size may be smaller for some of the species. That will be a combination of temperature and available nutrition. We're looking at more frequent encounters between predators and prey. And then, of course, overlaying this, we have longer lasting, more frequent, and more severe heat waves, which we know should reduce plant growth. We should see higher mortality, slower development, and reproduced reproduction in at least some of the species in these agricultural food webs, which then contradicts or contrasts against some of these warmer temperature effects. So life's complicated. Now, what I'm going to try and do is I'm going to try and convince you that we can use greenhouses as a model for future climates, and to try and start to tease out some of these effects. And what I'm going to talk about is a holistic experiment. It's one of those horrifying experiments that graduate students should never do. So in our greenhouses, we've got a carbon dioxide enrichment available. A lot of growers habitually use carbon dioxide supplementation in these big glass boxes. That's just how they grow. We tend to run average temperatures above ambient, because why else would you be using a greenhouse except to keep the plants and the system warm? And sunlight, solar effects, can lead to very high temperatures in greenhouses. The old greenhouse effect, as it were. So we can think about using greenhouses as models to try and tease out some effects. So I had available 2 compartments, and 10 in our greenhouses at Agassiz, 10 walk-in cages in each, and these are the walk-in cages in the background here. We set the one greenhouse at 400 parts per million, essentially ambient CO2. The other greenhouse at 800. We set the temperature in the high temperature, the future climate regime. I'll call it the high CO2 regime. So we set that about 4 degrees higher. We set the night cooling about 2 degrees higher. And we imposed a 1 to 1.5 degree ramp rate. So that...
A good question.
Yeah.
What's the level of CO2 that commercial growers?
Anywhere from 1,000 to 1,200 parts per million. So this is fairly modest compared to what a commercial grower would do. So yeah, they're really boosting the CO2. So yeah, we have a 1.5 degree ramp rate, which means that we don't allow the greenhouse to respond in an out of control fashion. The greenhouse computer limits the rate at which temperature can increase, so we get something that's more realistic, more comparable to an outdoor situation. And then my greenhouse manager came up with this brilliant idea to use a function in the computer which is called a light boost, which says when days are really bright, you let the greenhouse get hot, because then the plants can take advantage of the extra light units. And so we set a light boost at 350 watts per meter squared accumulated light during the day, which means that the greenhouse is then allowed to go 6 degrees above our otherwise set temperatures, generating our heat outbreak, our heat wave. These are just some typical 24-hour temperatures. You can see we kind of achieved what we set out to do. This is the lower line is the cooler environment, the high CO2 environment. You can see it was crossing a 32-degree threshold fairly frequently, depending again on how much light was available. This was a bright day. This was not such a bright day. And I was able to conduct 3 runs of an experiment over time in these greenhouses. The 32-degree threshold is important because that's the upper development threshold for green peach aphid. That's the line at which we decided in this system that constitutes heat wave. So we did pretty well. We hit our 400 parts per million target in each of the runs, got pretty close to 800 parts per million in the high CO2 regime. Temperatures were running about 4 degrees higher in the first run, a little closer together in the second run, and then well separated again in the third run. So we got these very different climate regimes, high CO2 and a low CO2 regime. Okay. I show you this. This is the cumulative growing degree days with a 10-degree base. The upper line is the high CO2 greenhouse without a cutoff available. That's essentially the heat units available for Gillespie carrier. The middle line is the high carbon dioxide house with a 32-degree upper threshold cutoff. That's the heat units available to green peach aphid. And then the solid line on the bottom is the low CO2 house with no cutoff. And since they rarely reach 32 degrees, we rarely had to impose that growing degree day cutoff rule. So we conducted this experiment with peppers, Mysis green peach aphid, Aphidius matricariae. In each house we had 2 cages with no aphids. That provided an index of the effect of the system on plant growth. 2 cages with aphids only, which then provided an index of the effect of aphids on plant growth in the 2 regimes, and then 6 cages which had the interesting stuff, which is the parasitoids and the aphids. We had 24 plants in each cage, selected 8 of those and destructively sampled them in each of weeks 3, 4, and 5. Slightly longer term experiment than our earlier ones. This is the early setup. Green peach pepper seedlings. As soon as they germinated, had the first true leaf, we popped an aphid on, left them for a week, and then put in 12 Aphidius matricuridae females in the cage to let things kind of cook. We took plant height, dry weight of the aphids, the plants, the aphid parasitoids. We measured carbon-nitrogen ratios. On 4 plants, we took a leaf area measurement, counted the aphids, the ANAs, the mummies, the parasitism rates on the 4th leaf from the cotyledon, using that leaf because the plants were varying so much in growth rate that that 4th leaf was the only one we were sure would be available. Carbon-nitrogen ratios held to the theory. We got a 10 to 20% split in the carbon-nitrogen ratio on plants without aphids. With parasitoids, the split was very similar. But when we added aphids, something really complicated happened. I only have 1 run available. We're still waiting to get the results on the last 2 runs, so I don't want to stick my neck out too far that there's an aphid effect on carbon-nitrogen ratios, but it's kind of intriguing. Predictably, but being an entomologist and not a plant physiologist, surprising to me, there was quite a difference in plant growth. The early run, so this run was conducted in February and March, this one in April and May, and this one in September and October. Light levels varied quite dramatically, and that generated quite a difference in plant growth weight, or plant dry weight, in the 3 different runs. This is the week 5 only. But what you see is that there's always a split between the high carbon dioxide and the low carbon dioxide houses. There's always an effect of aphids, which is quite dramatic relative to the no aphid index plants, and the parasitoid plants with aphids and parasitoids are always intermediate between the no aphid and the aphid plants. So great. With all this variation, how do you think about this? And the way to think about this is to think about it as think of plant weight as a fraction of the plant in the no aphid controls. So plants in the no aphid controls are doing about as good as they possibly can. If you look at the plant growth in aphid-only cages, so there's no parasitoids here, as a fraction of the plants without aphids, the relative effect is identical. There's absolutely, so plants are going to be impacted by aphids, but the fact that they're being impacted by aphids in high carbon dioxide regimes doesn't really matter all that much. The relative amount of plant loss is the same irrespective of future climate or present climate. When you add the parasitoids, something quite different happens. So you have about a 30% increase in the proportion of plant growth that is recovered as a result of the high carbon dioxide, in the high carbon dioxide regime, as a result of parasitoid foraging. So this is parasitoid effect on the trophic cascade. And what you see is a dramatic increase in the amount of plant tissue that is retained in the system as a result of the foraging of the effects of the parasitoids on aphid population. Parasitism rates increased quite differently in the 2 houses. So we have parasitism rates starting out when we started to measure them much higher in the high carbon dioxide house than the low carbon dioxide house. Probably quite predictably, both peaked very close to 100% parasitism. The high carbon dioxide house has started to drop. The low carbon dioxide house was staying high by week 5. So this is one of those situations you look at the graph and you say, dang, I wish I'd let that go another week. But, hindsight's always 20/20 in this kind of experiment. Aphid dry weights, I don't have the dry weights for the last run yet, but what you can see is something kind of interesting. Dry weight when we first, in week 3 was 0.1 grams of aphids, increased to about 0.3 grams of aphids, and then dropped quite dramatically. In the context of Jordan Banham's models, again, I wonder if I'd be leaving this for a couple of extra weeks, would we have seen the population start to go extinct in those cages, where we actually would we have seen the effect that Jordan was predicting in the context of heat waves. Conversely, in the first run, aphid populations were continuing to increase. The second run, quite disappointingly, was very different. The two populations started out very much the same, increased to more or less the same threshold, and then aphid weights were decreasing somewhat less slowly, but I'm not sure if they were significant yet or not yet. I'm not going to start trying to tease this apart until I have the third run available. But you can see that there's, again, a trend to decreasing aphid populations. Of course, I cheated. Those first two graphs were on very different scales. This is the two runs on the same scale. The first run, aphid populations were a lot lower overall, and in the second run, they were much higher. And I think there's probably some really interesting effects of average temperature and temperature thresholds going on in this system. What's happening in this first run is the greenhouse in the low temperature regime never exceeded 32 degrees Celsius, so it never went over the development threshold of the aphid. The high CO2 house stayed quite routinely. Conversely, in the second run, in the middle of the summer, the cool greenhouse was actually going over the 32 degree threshold quite routinely. That's the average average maximum temperature. So, which might, because these are much closer together, might explain why these two plots are much closer together. And it also suggests that the upper development threshold was playing some kind of a role in the shape of the community in general. So, under these simulated future climate conditions, we were affecting plant size and leaf area. Plant growth in particular was different. Carbon-nitrogen ratio increased. Plants may have supported greater numbers and higher densities of green peach aphid. I didn't show you the density graph, but it looked like there were more aphids per unit area in the high CO2 environment, which would make sense because if plants have a higher carbon-nitrogen ratio, aphids have a harder time getting at nutrition from the plant. They would very likely be smaller. And again, this is one of those things where I take a graduate student, smack them on the back of the head, or her. No, I get to smack the guys, but not the girls. Because I didn't measure size of aphids. One of those things where I finished the experiment. And clearly, we're going to have to backtrack and see what is the effect of that high carbon regime on peppers on the effect of aphids. There's clearly going to be an effect. I'm almost certain. The trophic cascade was relatively larger in the future climate regime. Per capita value of biological control, as a result, was probably higher. That investment in 12 parasitoids got you a much better return in terms of plant dry weight than it did than under the low carbon dioxide regime. So you're getting a little bit better bang for your buck out of a biological control investment. And I think temperature range might be associated with the likelihood of extinction of local populations. Let's say that properly. Okay, again, go back to mechanisms. High carbon dioxide changed leaf size and plant size. If you change leaf size, you're changing the size of the arena under which predator-prey interactions take place. Search rate on a leaf that is twice the size means something quite different. So the arena has changed in nature. Nutrition and defense compounds were likely changed. Under high carbon dioxide, the salicylic acid pathway, it's one of the defense compound pathways in plants, is upregulated. And the jasmonic acid pathway, which is the chewing insect defense pathway, tends to be downregulated. It turns out that the salicylic acid pathway is one of the major defense pathways for defense against sucking insects like aphids and leafhoppers and scales. So is there something going on with defense compounds? Carrying capacity of the plant seemed to change, perhaps as a result of leaf size. There were interactions between individuals that were quite different and may have been quite different as a result of leaf size. Patch value, intraspecific competition. I think it begs us going back into the greenhouse to ask how the predators are looking at and valuing patches. Is there some difference in intraspecific competition? Temperature and development rates. Temperature clearly affects development rates, which affects generation times. It also affects foraging speed. It probably affects the fitness costs, the cost of avoidance, the cost of dispersal, the cost of leaving a suitable patch as an aphid and moving to a protected location to avoid damage from high heat. And the variation in range, the heat wave, affected relative degree day accumulations and was likely having an effect on parasitism rates. So there's a whole bunch of, so there's no answers in this slide. There's only a lot of questions. And I think what I'd like to suggest is that studies like this, somewhat better planned, somewhat better thought out, controlled in greenhouses with carbon dioxide control, give us an opportunity to start asking questions about how this future climate regime is going to impact biological control and integrated pest management systems that we rely on in agriculture. Keeping in mind, I said something along the line of the value of biological control was increased. I don't want to mean that. I don't want to say that as a generalized statement. It's specific to peppers, green peach aphid, Aphidius matricariae in cages in the greenhouses I was studying. Just all I'm saying is that climate change with high CO2 is likely going to change the effect of the trophic cascade, the impacts of biological control. It's not always going to change them in the same direction. I have to guess that development thresholds, upper development thresholds of the predators and prey are going to have a big impact on the nature of that interaction. Finally, just as a kind of wrap-up statement, climate change is this. There's a lot of reviews out there in the literature now, a lot of good reviews, and all of them suggest more or less the same thing when it comes to climate change and beneficial arthropods in agriculture. These beneficial arthropods are going to be disrupted by these future climate regimes. It's going to be highly idiosyncratic. It's going to depend on which natural enemy you're talking about. Without intervention, these are going to result in production losses, and those losses are likely going to justify active interventions. If those are pesticides, then a lot of our beneficial arthropod work that we've been doing over the last several decades is simply going to fall apart. So I will leave you with some thank yous. Bernie Reutberg, of course, Michael mentioned I worked with Bernie quite extensively, and Jordan Bannerman, the grad student, did a lot of the modeling work. Aphid Nazreen, a postdoc, who worked with me. David Eric, who is my plant physiologist, who shall take no blame whatsoever for my silly statements about plant physiology. Peggy Clark and Shoving Yu, my two technicians. My wife, Beth, some graduate students and co-op students who spent a lot of time counting aphids and giving me a hand in the lab. Jason Peason, the greenhouse tech, who has been really influential in developing this this climate regime and the people in the Agassiz greenhouse group and the Reutberg lab in general. Big thank you to all of these folks who helped bring a lot of this work to the point it's at now, and of course the funding agencies, including my employer. So, with that, questions?
Okay, let's thank Dave. I'm sure there'll be many questions, so please.
Okay, so thinking about, you made the point about using this as a sort of a microcosm or microcosm for understanding climate change and interaction. One thing I wasn't hearing you talk about was sort of the nutrient status of the plants in terms of fertilizing them, what the basis of it is,
Right.
and whether you think that sort of in the greenhouse, I mean, presumably you were fertilizing them in some industry standard.
Yeah.
So first step, then sort of if you were speculating about how things might be different in a natural system where, again, you said nitrogen is limiting terrestrially, do you expect there to be a more dramatic interaction in terms of the forms of aphids?
I would expect so, for two reasons. One is that I think a lot of, well, in agriculture, though, we tend to be pretty generous with nitrogen, in fact, overly generous. What I think will be limiting is nitrogen that is free and available to be absorbed by the plant, which is a function of water. So as soon as, and that's one of the climate change effects that's going to be really uncertain. How is water going to be affected? Rainfall, presumably is going to change. There have been some measurable changes in the frequency of high rainfall environment, high rainfall events in certain places. Is drought going to be associated with climate change? There's a lot of really big system questions that I think the modelers haven't got to yet. But my guess is if you go into the natural environment, drought and water availability is going to play a huge role. And as plants begin to shut down transpiration as a result of having water restrictions, you're going to see restrictions in growth, you're going to see changes in nitrogen uptake, you're going to see some fairly dramatic changes in methane. So, for example, plants that are restricted with respect to water are going to stop defending themselves by cooling themselves by transpiration. There's a lot of really, really weird things starting. Leaves start to wilt, plants will wilt. And that generates changes in salicylic acid pathways. So yeah, it's really complicated. But I think systems like this give us an opportunity to start asking some questions about how those factors of drought are going to play out in the context of all the other stuff that's going on in the context of climate change. Is that sort of
Yeah, so I mean, you highlight water. So you think the water effect will trump the nitrogen effect?
Trump? No, competent. Yes.
Okay. No other questions? Let's thank Dave. System. So, for example, a lot of how we use Mirity in greenhouses, how we do that really has come out of Dave's lab. He was elected as an honorary member of IOBC, I think, because of his work. And today he's going to be talking about a broader topic, that is the influence of climate change on biological control. He is also an adjunct professor at Simon Fraser, who works very closely with Bernie Reutberg, that many people know. And I think Bernie's a little bit of inherent in this presentation as well. So with that, Dave.
Oh, great. Thank you, Michael. It's a tremendous honor and pleasure to be down here, and coming from British Columbia, where the temperatures are running in the 40s. If I'm lucky, daytime highs, and it's pouring rain. This is lovely weather, 20 degrees, and no rain. It's a real treat. So yeah. Well, you've been hoping for rain, so maybe you'll bring it with me. Yeah. So this what I'm going to talk about today sort of stems from work that I started a number of years ago on biological control of aphids in greenhouse peppers. And we developed this little predator-prey parasitoid host system with green pea Jacob that we were starting to try and tease apart. Why does it work? How does it work? When an opportunity came along to start to think about in the context of perturbations and the influence of high temperatures on this system. So what I'm going to talk about today is some of that work where it started from what we found initially, and then some studies that are very much still in the wet ink stage. I've just finished collecting the last little bit of data. In fact, my technicians are still processing. A lot of a future with increased mean temperatures is going to hold the increased probability of heat waves. The heat waves will be longer, they will be more frequent, and they'll be more intense. And then that begs the question, well, why do we care? And the simple answer is, in part, in this universal relationship between temperature and a host of life history and biological phenomena. Here I've just graphed development rate as a function of temperature, something every entomologist is familiar with. We have a lower development threshold on the x-axis, a temperature below which the process doesn't take place in net effect. And then as temperature increases, the rate of the process increases to some upper threshold, at which point it's going about as fast as it possibly can. Continue to increase the temperature, and we reach an upper development threshold, a point above which the process, whatever it is, doesn't take place in net effect. If you think about that, extend that x-axis, you have a lower critical temperature, that is a temperature below which the phenomena, the organism freezes or dies from accumulated cold injury, and then at the upper end, around 40-something degrees, you have an upper critical temperature, a temperature at which enzymes denature, the animal dies. So those relationships are really, really important in biology. I've showed development rate. Keep in mind, and so this range in here between the upper threshold and the upper critical temperature is the one that's really important in the context of heat waves. Getting ahead of myself here, some of the samples today, so it's going to be very recent work, which is very much unfinished, soggy clay, as it were. Biological control in the face of climate change. Everybody's presumably seen this graph or an iteration of it. Carbon dioxide levels in the atmosphere are changing. They're changing dramatically in post-industrial revolution time. And as a consequence, we see this phenomenon called global climate change. An increasing concentration of carbon dioxide, which is leading to changes in ocean pH, changes in global average temperatures, changes in wind patterns, changes in rain patterns, and so on and so forth. And there's a sort of in the public consciousness, there's this global trend towards increasing temperature, and every time you get a cold day, you get the naysayers sticking up their hands. But those increasing global mean temperatures are changing the world around us in a fairly profound way. We're seeing changes in distributions and ranges of animals, changes in seasonal activity, synchrony between parasitoids and hosts, flowers and pollinators. We're seeing changes in overwintering survival. So we're seeing a whole host of things happening as a result of this increasing mean global temperature. But there's another trend underlying that, which is that as that global mean temperature changes, there's a tendency towards increasing variation around that mean temperature. So you're getting greater and greater extremes of temperature. And the models tell us then that the future is going to hold a...
My pleasure to be able to introduce our speaker for today, Dr. David Gillespie from the Pacific Agricultural Research Center in beautiful Agassiz, British Columbia. And for those who've been up there, it is an absolutely beautiful station. I was very lucky about 20 years ago. I spent a short sabbatical with Dave and I had a wonderful experience there. So if you're ever up in that area, it is really spectacular. Dave is a native Canadian. Obviously, actually spent most of his time in British Columbia as both a actually his Bachelor of Science degree in biology, a Master's in biology, and his PhD in biology from Simon Fraser University. So he didn't go too far away, basically, and now he's at Agassiz, where he is a senior research scientist. So Dave's area primarily is biological control almost exclusively. He does work in sort of field systems and with classical biological control. He has a large project now on canola, for example. But his major contribution really is focused on biological control in greenhouse systems. For those that know British Columbia, the greenhouse industry there is huge. The vegetable industry especially, so Dave has made remarkable progress in advancing biological control in vegetables, not only in BC, but literally around the world. And so what Dave has done is really, investigating sort of native natural enemies, understanding them, and basically show how they can be used in greenhouses. And so because of that effort, a lot of what we deal with now on a commercial basis, in other words, what's available commercially, internationally, really came out of Dave's lab. So predatory mites, some predatory cystocomyces, and especially the predatory murids. A lot of that work, a lot of biocontrol that goes on in California, but those natural enemies, actually, the foundation of that work has come out of Dave's lab. And not only has he developed new natural enemies, but sort of the delivery.
So that's the zone in which you see accumulated physiological stress. You see accumulated damage. You see organisms starting to change their behavior in response to these threatening conditions. So there's a lot of stuff taking place in this zone, which is the temperature at which we see, which is of concern for these heat wave environments. And I mentioned, showed development rate, walking speed increases as a function of temperature. Predation can have a neat little curvilinear function as a result of temperature. So all kinds of things change in an organism's biology as a function of the temperature at which it's foraging at. Body size. And that's a well-known relationship. As temperature increases, body size tends to increase, again, in some kind of a curvilinear fashion. That's great. But smaller organisms eat that stuff. So we've been studying this phenomenon of heat waves and high temperature effects in this community that I've been working with, Aphelinus abdominis. So it's based in sweet pepper. It's a greenhouse crop I work with. Green peach aphid as the host, and then two parasitoids, Abraconid, Aphidius matricariae, and an aphelinid, Aphelinus abdominalis. And so we just, in case you're not completely familiar with this system, green peach aphid takes 7 to 8 days to go from immature first instar to adult or viviparous. It's a great little animal to work with. The parasitoid